Hello, welcome to our program. Dear students, today we are going to talk about the topic selective permeability of the membranes, membrane transport, liposomes, freeze etching and freeze fracture technique for membrane study. Now let's first talk about the objectives that we'll be having. To study the selective permeability of the membranes, to discuss the membrane transport, to study the detailed account of active transport, to discuss different types of diffusion, to have a detailed account of liposomes, to understand the freeze etching and freeze fracture technique for membrane study. Dear students, let's begin with selective permeability of the membranes. The plasma membrane is a selectively permeable barrier between the cell and the extracellular environment. Its permeability properties ensure that essential molecules such as ions, glucose, amino acids and lipids readily enter the cell, metabolic intermediates remain in the cell, and waste compounds leave the cell. Thus, the selective permeability of the plasma membrane allows the cell to maintain a constant internal environment. Movement of virtually all molecules and ions across cellular membranes is mediated by selective membrane transport proteins embedded in the phospholipid bilayer. Because different cell types require different mixtures of low molecular weight compounds, the plasma membrane of each cell type contains a specific set of transport proteins that allow only certain ions and molecules to cross. Similarly, organelles within the cell often have different internal environment from that of surrounding cytosol, and the organelle membranes contain specific proteins that maintain this difference. The phospholipid bilayer, the basic structural unit of biomembranes, is essentially impermeable to the most of water-soluble molecules, ions, and water itself. If a transported substance carries a net charge, its movement is influenced by both its concentration gradient and the membrane potential, the electric potential that's voltage across the membrane. The combination of these two forces, called the electrochemical gradient, determines the energetically favorable direction of transport of a charged molecule across a membrane. The electric potential that exists along most cellular membranes results from a small imbalance in the concentration of positively and negatively charged ions on the two sides of the membrane. Few molecules cross membrane by passive diffusion. Gases such as oxygen and carbon dioxide and small uncharged polar molecules such as urea and ethanol can readily move by passive diffusion across an artificial membrane composed of pure phospholipid or a phospholipid and cholesterol. The diffusion of molecules across cellular membranes without the aid of transport proteins and metabolic energy down its chemical concentration gradient is known as passive diffusion. The relative diffusion rate of any substance across pure phospholipid bilayer is proportional to its concentration gradient across the layer and to its hydrophobicity and size. Charged molecules are also affected by any electric potential across the membrane. Membrane proteins mediate transport of most molecules and all ions across biomembranes. Very few molecules and no ions can cross a pure phospholipid bilayer at appreciable rates by passive diffusion. Thus, transport of most molecules into and out of cells require assistance of specialized membrane proteins. Even transport of molecules with a relatively large partition coefficient, that's water and urea, is frequently accelerated by specific proteins because their transport by passive diffusion usually is not sufficiently rapid to meet cellular needs. All transport proteins are transmembrane proteins containing multiple membrane-spanning segments that generally are alpha helices. By forming a protein-lined pathway across the membrane, transport proteins are thought to allow movement of hydrophilic substances without their coming into the contact with the hydrophobic interior of the membrane. Dear students, now let's discuss in detail the membrane transport. First, we'll talk about facilitated diffusion. Channel proteins transport water or specific types of ions and hydrophilic small molecules down their concentration or electric potential gradients. Such protein-assisted transport sometimes is referred to as facilitated diffusion. Channel proteins form a hydrophilic passageway across the membrane through which multiple water molecules or ions move simultaneously single file at very rapid rate. Some ion channels are open much of the time. These are referred to as non-gated channels. Most ion channels, however, open only in response to specific chemical or electric signals. These are referred to as gated channels. Facilitated diffusion differs from simple diffusion in many ways. First, the rate of simple diffusion increases proportionately with the concentration of the diffusing substance. Second, in the facilitated diffusion, the rate of diffusion approaches maximum as the concentration of the substance increases. This maximum rate is dictated by the rate at which the carrier protein molecule can undergo the conformational changes. Now let's have a look at active transport. ATP-powered pumps, or simply pumps, are ATPases that use the energy of ATP hydrolysis to move ions or small molecules across a membrane against a chemical concentration gradient or electric potential of both. This process, referred to as active transport, is an example of a coupled chemical reaction. In this case, transport of ions or small molecules uphill against an electrochemical gradient, which requires energy, is coupled to the hydrolysis of ATP, which releases energy. The overall reaction, ATP hydrolysis, and the uphill movement of ions or small molecules energetically favorable. 4 classes of transmembrane proteins couple the energy-releasing hydrolysis of ATP with the energy-requiring transport of substances against their concentration gradient. Those are P, V, and F class pumps and ABC proteins. The combined action of P class sodium ion potassium ion ATPases in the plasma membrane and homologous calcium ions ATPases in the plasma membrane or sarcoplasmic reticulum creates the usual ion milieu of animal cells, that is, high potassium ion, low calcium ion, and low sodium ion in the cytosol, low potassium ions, high calcium ions, and high sodium ions in the extracellular fluid. In P-class pumps, phosphorylation of the catalytic subunit and a change in conformational states are essential for coupling ATP hydrolysis to transport hydrogen ions, sodium ions, calcium ions, and potassium ions. V and F class ATPases, which transport protons exclusively, are large multi-subunit complexes with a proton conducting channel in the transmembrane domain and ATP binding sites in the cytosolic domain. V class H pumps in animal lysosomal and endosomal membranes and plant vacuole membranes are responsible for maintaining a lower pH inside the organelles than in the surrounding cytosol. All members of the large and diverse ABC superfamily of transport proteins contain 4 core domains, that is, 2 transmembrane domains which form a pathway for solute movement and determine substrate specificity, and 2 cytosolic ATP binding domains. The ABC superfamily includes bacterial amino acid and sugar permeases and about 50 mammalian proteins. For example, transporters, also called carriers, move a wide variety of ions and molecules across cell membranes. Active transport can be defined as the movement of molecules across a cell membrane from a region of low concentration to a region of high concentration in the direction against concentration gradient. Active transport depends upon the integral membrane proteins that selectively bind a particular solute and move it across the membrane in a process driven by the change in the protein's conformation. Movement of a solute against a gradient requires the coupled input of energy. Consecutively, the endergonic movement of ions or other solutes across the membrane against a concentration gradient is coupled to an exergonic process such as hydrolysis of ATP, the absorbance of light, the transport of electrons, or flow of other substances down their gradients. Proteins that carry out active transport are often referred to as pumps. Dear students, now let's discuss first active transport driven by ATP hydrolysis. In 1957, Jens Sakow, a Danish physiologist, proposed that the ATP hydrolyzing enzyme that was only active in presence of both sodium and potassium ions in nerve cells of crab was the same protein that was active in transporting the 2 ions. The enzyme was called sodium ion potassium ion ATPase or the sodium potassium pump. In active transport, the energy provided by the coupled reaction is used to drive the uphill transport of molecules in the energetically unfavorable direction. In other words, for each ATP hydrolyzed, 3 sodium ions are pumped out as 2 potassium ions are pumped in. Because of this pumping ratio, the sodium-potassium ATPase is electrogenic, which means to contribute directly to the separation of charge across the membrane. This pump is an example of a P-type pump, where P stands for the phosphorylation. The concentration of sodium ions is approximately 10 times higher outside than inside of the cells, while the concentration of potassium ions is higher inside than out. These ion gradients are maintained by sodium-potassium pump, which uses energy derived from the ATP hydrolysis to transport sodium ions and potassium ions against their electrochemical gradients. This process is a result of ATP-driven conformational change in the pump. First, sodium ions bind to the high-affinity sites inside the cell. This binding stimulates the hydrolysis of ATP and phosphorylation of the pump, inducing a conformational change that exposes a sodium ion binding sites to the outside of the cell and reduces their affinity for sodium ions. Consequently, the bound sodium ions is released into the electrocellular fluids. At the same time, high-affinity potassium ions binding sites are exposed on the cell surface. The binding of extracellular sodium ions to these sites then stimulates the hydrolysis of the phosphate group bound to the pump, which induces a second conformational change, exposing the potassium ion binding sites to the cytosol and lowering their binding affinity, so that the potassium ions is released inside the cell. The pump has 3 binding sites for sodium ions and 2 for potassium ions, so each cycle transports 3 sodium ions and 2 potassium ions across the plasma membrane at the expense of 1 molecule of ATP. The importance of the sodium-potassium pump is indicated by the fact that it's estimated to consume nearly 25% of the ATP utilized by many animal cells. One of the critical roles of the sodium ions and potassium ion gradients established by the pump is the propagation of electric signals in the nerve and muscles. Dear students, now let's discuss some other ion transport systems. First is calcium ATPase. The best studied P-type pump is the calcium ATPase, whose 3-dimensional structure has been determined at several stages of the pumping cycle. The calcium pump is present in the membranes of the endoplasmic reticulum, where it actively transports calcium ions out of the cytosol into the lumen of this organelle. The transport of the calcium ions by the calcium ATPase is accompanied by large conformational chains which couple the hydrolysis of ATP to change in access and affinity of the ion binding sites. Hydrogen-potassium ATPase. The epithelial lining of the stomach contains potassium-hydrogen ATPase, which secretes solution of concentrated acid up to 0.16 normal of HCl into the stomach chamber. In the resting state, these pump molecules are situated in the cytoplasmic membranes of the parietal cells of the stomach lining and are non-functional. When food enters the stomach, hormonal message is transmitted to the parietal cells that causes the pump-containing membranes to move to the apical cell surface, where they fuse with the plasma membrane and begin secreting acid. Third, hydrogen transporting. Plants have a hydrogen transporting pump. This proton pump plays a key role in the secondary transport solutes in the control of cytosolic pH and possibly in the control of cell growth by means of acidification of the plant cell wall. Dear students, now let's discuss the active transport done by the light energy. Halobacterium salinarium is an archaebacterium that lives in the extremely salty environments such as that found in the Great Salt Lake. When grown in anaerobic conditions, the plasma membranes of these prokaryotes take on a purple color due to the presence of one particular protein, bacteriorhodopsin. This protein contains retinal, the same prosthetic group present in the rhodopsin, the light-absorbing protein of the rods of the vertebrate retina. The absorption of light energy by the retinal group induces a series of conformational changes in the protein that causes a proton to move from the retinal group through a channel in the protein to the cell exterior. The proton donated by the photoexcited retinal is replaced by another proton transferred to the protein from the cytoplasm. In effect, this response results in the translocation of the proton from the cytoplasm to the external environment, thereby generating a steep H-positive gradient across the plasma membrane. This gradient is subsequently used by an ATP synthesizing enzyme to phosphorylate ADP. Now let's discuss co-transport, coupling active transport to existing ion gradients. The establishment of concentration gradients such as those of sodium ions, potassium ions and hydrogen ions provides a means by which free energy can be stored in a cell. The potential energy stored in ionic gradients is utilized by a cell in various ways to perform work, including the transport of other solutes. Consider the physiological activity of the intestine. Within its lumen, enzymes hydrolyze high molecular weight polysaccharides into simple sugars, which are absorbed by the epithelial cells that line the intestine. The movement of glucose across the apical plasma membrane of the epithelial cells against a concentration gradient occurs by the co-transport with sodium ions. The sodium ion concentration is kept very low within the cell by the action of primary active transport system, that's sodium-potassium ATPase, located in the basal lateral plasma membrane, which pumps sodium ions out of the cell against a concentration gradient. The tendency for sodium ions to diffuse back across the apical plasma membrane down their concentration gradient is tapped by the epithelial cells to drive the co-transport of the glucose molecules into the cell against the concentration gradient. The glucose molecules are said to be driven by secondary active transport. In this case, the transport protein called a sodium-glucose co-transporter moves 2 sodium ions and 1 glucose molecule with each cycle. Once inside, the glucose molecules diffuse through the cell and are moved across the basal membrane by facilitated diffusion. Dear students, now let's have a brief idea about the liposomes. Liposomes are small artificial vesicles of spherical shape that can be created from the cholesterol and natural non-toxic phospholipids. An important feature of lipid bilayer is its ability to reassemble, which can be demonstrated more easily within a test tube than a living cell. If a small amount of phosphatidylcholine is dispersed in an aqueous solution, the phospholipid molecules assemble spontaneously to form the walls of fluid-filled spherical vesicles called liposomes. The walls of these liposomes consist of a continuous lipid bilayer of a natural membrane. Liposomes have proven invaluable in membrane research. Membrane proteins can be inserted into the liposomes and their function studied in a much simpler environment than that of a natural membrane. Liposomes have also been developed as vehicles to deliver drugs or DNA molecules within the body. The drugs and the DNA can be linked to the wall of the liposome or contained at high concentration within its lumen. In these studies, the walls of the liposomes are constructed to contain specific proteins such as antibodies or hormones that allow the liposomes to bind selectively to the surfaces of particular target cells where the drug or DNA is intended to go. Most of the early clinical studies with liposomes met with failure because the injected vesicles were rapidly removed by phagocytic cells of the immune system. This obstacle has been overcome with the development of so-called stealth liposomes, for example, calyx, that contain an outer coating of synthetic polymer that protects the liposome from immune destruction. Now let's discuss the clinical applications of the conventional liposomes. Based on these studies, conventional liposomes composed of neutral or negatively charged lipids plus cholesterol have been prepared. Some of these formulations have reached the market or are now entering clinical trials. Ambisome, in which the encapsulated drug is the antifungal amphotericin B, Myoset, encapsulating the anticancer agent doxorubicin, and Donosome, in which the drug incorporated is donorubicin, are the principal examples of such formulations. To obtain stable formulations incorporating a constant amount of drug, various mechanisms are exploited. Ambisome is lyophilized, Myoset is supplied in 3 separate vials, one containing doxorubicin as dry powder, one a solution of empty lysosomes in citric buffer, and the third a solution of sodium carbonate. In this case, drug entrapment must be achieved immediately prior to the administration. Donosome is at present the only pure lipid MPS-avoiding liposomal formulation. It is available as a stable, ready-to-inject liposomal formulation. By addition of sphingomyelin and saturated fatty acid chain lipids to the lipidic formulation, 2 commercial liposome formulations have been produced. A novel liposomal formulation of vincristine based on the sphingomyelin cholesterol unilamellar vesicle has recently been shown to be efficacious in the treatment of relapsed non-Hodgkin's lymphoma. Inex Pharmaceutical Company has developed 2 further candidate formulations, that is INX-0125, which is a liposomal vinorelbine, and INX-0076, which is liposomal topotecan, also based on the use of sphingomyelin cholesterol mixture. Another liposomal formulation composed of hydrogenated soy phosphatidylcholine and cholesterol and containing lototecan has been developed and named OSI-211. Clinical results have shown that incorporation of OSI-211 in the acid aqueous core of the vesicle, in addition to providing the known therapeutic advantage of a liposomal carrier, can also favor maintenance of lactone ring closure of lototecan, the active form, which increases stability of the active compound, consequently improving its tumor toxicity. Two other liposome formulations employing saturated phospholipids have been launched for clinical development. These are Niotran and Eroplatin. These are multilamellar liposomal formulations consisting of dimyristoyl phosphatidylcholine and dimyristoyl phosphatidylglycerol. Niotran contains nystatin A1 and a membrane-active polyene antifungal antibiotic that is structurally related to amphotericin B. Eroplatin is a multilamellar liposomal formulation of cis bis neodecanoto trans RR12 diaminocyclohexane platinum 2, a hydrophobic structural analog of oxaliplatin. Eroplatin is in clinical trials for a wide range of tumors and more recently for advanced solid tumors and B-cell lymphoma. Aeronex Pharmaceuticals has also developed a liposomal all-trans retinoic acid formulations, also known as Atrogen, containing tretinoin, dimyristoyl phosphatidylcholine, and soybean oil. The formulation has shown efficacy in the treatment of acute promyelocytic leukemia and other retinoid-responsive cancers. Several interesting liposome preparations have been developed by Neopharm. In LEM-ETU, mitozentrone is encapsulated in multilamellar liposomes composed of di-oleophosphatidylcholine and cholesterol after charge interaction with the cardiolipin. LEP-ETU, a liposome-entrapped, easy-to-use paclitaxel formulation, recently demonstrated bioequivalence with Taxol and interesting activity in phase 1 trials. This formulation, composed of di-oleophosphatidylcholine, cholesterol, and cardiolipin, is capable of carrying paclitaxel in the liposome bilayer at a maximum mole percent of about 3.5%. In LESN38, the active metabolite of irinotecan, which is 7-ethyl-10-hydroxycamphothecin, is encapsulated in phosphatidylcholine, cholesterol, and cardiolipin liposomes. Paclitaxel has also been encapsulated in other modern formulations of cationic lipid complexes MBT0206 that have been shown to be bounded and internalized selectively by angiogenic tumoral endothelial cells after intravenous injection. A phase 2 clinical trial is in progress for candidate drug ENDO-TAG1 in the treatment of advanced pancreatic cancer. Cytarabine is an antimetabolite antineoplastic agent used in clinical application for acute lymphoid leukemia, myeloid leukemia, and meningeal leukemia. Depocyte is a slow-release formulation of cytarabine designed for intrathecal administration in the treatment of neoplastic meningitis due to the breast cancer. In Depocyte, cytarabine is encapsulated in the aqueous compartment of a spherical 20 micrometers matrix comprised of lipid biochemically similar to normal human cell membranes, that is phospholipids, triglycerides, and cholesterol, also called depofoam. Anamycin, which is 3'-D-amino 4'-epi 3'-hydroxy 2'-iodo 4-dimethoxydoxorubicin, is a second-generation highly lipophilic anthracycline developed by the MD Anderson Cancer Center. Its molecular design will be able to avoid the multi-drug resistance mechanism of tumor cells, and its lipophilicity produces increased encapsulation in liposomes. Liposomal anamycin was in phase 2 trials in late 2005 in patients with refractory or relapsed acute lymphocytic leukemia. Dear students, now lastly, let's discuss freeze fracture replication and freeze etching. These techniques are used to look at the membranes and reveal the pattern of the integral membrane proteins. The preparation of samples by freeze fracture in combination with metal shadowing has been particularly important in studies of membrane structure. Specimens are frozen in liquid nitrogen at -196 degrees centigrade and then fractured with a knife blade. This process frequently splits the lipid bilayer, revealing interior faces of a cell membrane. The specimen is then shadowed with platinum and the biological material is dissolved with acid, producing a metal replica of the surface of the sample. Examination of such replicas in electron microscope reveals many surface bumps corresponding to the proteins that span the bilayer. Small pieces of tissue are placed on a small metal disc and rapidly frozen. The disc is then mounted on a cooled stage within a vacuum chamber and the frozen tissue block is struck by a knife edge. The resulting fracture plane spreads out from the point of the contact, splitting the tissue into two pieces, not unlike the way that an X plate splits a piece of wood in two. These fractures tend to cause deviations in the fracture plane, either upward or downward, giving the fracture face elevations, depressions, and ridges that reflect the contours of the protoplasm transversed. Consequently, the surface exposed by the fracture contain information about the contents of the cell. The goal is to make this information visible. The replication process accomplishes this by using a fractured surface as a template on which a heavy metal layer is deposited. The heavy metal is deposited onto the newly exposed surface of the frozen tissue in the same chamber where the tissue was fractured. The metal is deposited at an angle to provide shadows that accentuate local topography. A carbon layer is then deposited on top of the metal layer to cement the patches of the metal into a solid surface. Once this cast has been made, the tissue then provided the template can be thawed, removed, and discarded. It is a metal carbon replica that's placed on the specimen grid and viewed in the electron microscope. Variations in thickness of the metal in different parts of the replica cause variations in the numbers of the penetrating electrons to reach the viewing screen, producing the necessary contrast in the image. Fracture planes take the path of the least resistance through the frozen block, which often carries them through the center of the cellular membranes. As a result, this technique is particularly well suited for examining the distribution of integral membrane proteins as they span the lipid bilayer. Such studies carried out by Daniel Branton and others played an important role in the formulation of the fluid mosaic structure of cellular membrane in the early 1970s. Freeze fracture replication by itself is an extremely valuable technique, but it can be made even more informative by including a step called freeze etching. In this step, the frozen fractured specimen, while still in place within the cold chamber, is exposed to a vacuum at an elevated temperature for 1 to 5 minutes, during which a layer of ice can evaporate from the exposed surface. Once some of the ice has been removed, the surface of the structure can be coated with a heavy metal and carbon to create a metallic replica that reveals both the external surface and internal structure of the cellular membranes. The development of deep etching techniques in which greater amounts of surface ice are removed led to a fascinating look at cellular organelles. The technique delivers very high resolution and can be used to reveal the structure and distribution of macromolecular complexes such as those of cytoskeleton, as they are presumed to exist within the living cell. Well, that's all we have in today's program. I really hope you would have understood. Thanks a lot.